Everything dies. You know that. I know that. The bacterium knows it. The tree knows it. The elephant knows it. You are born, you grow, you age, you fall apart, and you die. That is the deal. That is the arrangement. Physics says so. The second law of thermodynamics, the most ironclad, unchallengeable, unforgiving law in all of nature, says that disorder always increases. Everything runs down, everything decays, every structure, no matter how magnificent, will eventually crumble into dust. Stars die, galaxies dissolve. The universe itself is winding down like a clock with nobody left to wind it. So of course you die, of course I die, of course everything that breathes and moves and thinks must at last stop. That is completely wrong, and I can prove it. Not with some fancy philosophical argument, not with wishful thinking, or religion, or poetry, though all of those have had a go at the problem. No, with an animal, a real living observable animal that you can hold on the tip of your pinky finger, an animal that looked the second law of thermodynamics right in the face and said, "No thank you, I think I'll go the other way." And the infuriating thing, the beautiful thing, is that it's been doing this quietly in the ocean for longer than we've had the brains to notice. We built pyramids and wrote symphonies and split the atom, and the whole time this creature was floating 6 inches under the waves, doing the one thing we were absolutely certain couldn't be done. But before I show you this creature, I want to make sure you really feel the weight of the problem. Because if you don't understand why death seems unavoidable, you won't appreciate just how astonishing it is that something has found a way around it. I want you to feel it in your bones. I want you to be so convinced that death is mandatory that when I introduce the exception, it hits you like a punch. So let's start with physics. Let's start with what we think we know. You are an arrangement, that's all. You're an incredibly sophisticated, staggeringly complex arrangement of atoms. Carbon, hydrogen, oxygen, nitrogen, a smattering of phosphorus and sulfur, a few trace metals. Nothing special about the ingredients. You could find them in dirt. You could find them in the air. What makes you alive is not what you're made of, but how it's put together. The pattern, the organization, the fact that these atoms are not scattered randomly in a pile, but are woven into proteins and membranes and double helices and little molecular machines that pump and twist and fold and copy with a precision that would make any watchmaker weep with envy. Now, the second law of thermodynamics is essentially a statement about patterns. It says that in any closed system, the amount of disorder, what we call entropy, tends to increase over time. Things fall apart more easily than they come together. You can shuffle a deck of cards 1,000,000 times and you'll never accidentally get them back in perfect order. You can drop a coffee cup and it shatters into 100 pieces, but you will never see 100 pieces of ceramic leap off the floor and reassemble into a cup. The arrow only goes one way. Disorder increases, order decreases. That's the law. And it's not a weak law, it's not a suggestion, it's not like a speed limit that people occasionally ignore. Arthur Eddington, the great English astrophysicist, once said something I love. He said, if someone points out to you that your pet theory of the universe is in disagreement with Maxwell's equations, then so much the worse for Maxwell's equations. If it's found to be contradicted by observation, well, these experimentalists do bungle things sometimes, but if your theory is found to be against the second law of thermodynamics, I can give you no hope. There is nothing for it but to collapse in deepest humiliation. That's how certain we all were. That's how certain we all were. The second law is why your car engine produces waste heat. It's why a glass of hot water always cools down to room temperature and never heats up by itself. It's why every fire eventually goes out. It's why when you break an egg, you can't unbreak it. At least, not without doing an enormous amount of work that generates even more disorder somewhere else. The universe has a very clear preference, and the preference is for mess. Erwin Schrödinger, brilliant man, one of the founders of quantum mechanics, asked a wonderful question in 1943. He gave a series of lectures in Dublin, and he asked very simply, what is life? And part of his answer was this striking idea. He said a living organism maintains its organization, its exquisite low entropy state, by continually feeding on what he called negative entropy from its environment. It drinks orderliness. A living creature takes in food, which is structured, organized matter, extracts the order from it, and dumps the waste, the high entropy leftovers, back into the environment. That's how you stay alive. You are an entropy pump. You suck in order and spit out disorder. And as long as you can keep doing that, you persist. You maintain your improbable pattern against the relentless tide of the second law. Beautiful idea. And it's correct as far as it goes. But here's the trouble. Even the best pump eventually wears out. Even the most exquisite machine accumulates damage over time. And that brings us to the real question. Not why do we live, but why do we stop? Let me tell you about your cells. You are made of roughly 37 trillion of them, give or take a few trillion, and every single one carries a copy of your genetic instructions, your DNA, coiled up inside the nucleus. When a cell needs to divide, it has to copy all of that DNA, every base pair, the whole enormous manuscript, billions of letters long, and the molecular machinery that does the copying is remarkably good. But it has a problem. At the ends of your chromosomes, there are these repeating sequences of DNA called telomeres. Think of them like the plastic caps on the ends of shoelaces. They don't carry any useful genetic information themselves. They're just protective buffers. And here's the trouble. Every time a cell copies its DNA to divide, the copying machinery can't quite reach the very end of the molecule. It's a geometric problem, really. The enzyme that does the copying needs to grab onto the strand a little way in from the tip, which means the last little bit doesn't get copied. So every time a cell divides, the telomeres get a tiny bit shorter. A tiny bit of the protective cap is lost. Now, for a while, this doesn't matter. The telomeres are long enough that losing a little each time is no big deal. But after enough divisions, about 40 to 60 for most human cells, the telomeres have gotten so short that they can no longer protect the chromosome properly. And at that point, the cell receives a signal, a very firm signal that says, stop dividing, you're done. The cell enters what we call senescence. It's still alive, still metabolically active, but it can't divide anymore. It sits there doing its job less and less well, accumulating damage, leaking inflammatory molecules into the surrounding tissue. A man named Leonard Hayflick figured this out in 1961, working at the Wistar Institute in Philadelphia. Before Hayflick, there was a kind of dogma in biology that cells were immortal. A famous surgeon named Alexis Carrel claimed in 1912 that he'd kept chicken heart cells alive and dividing in a dish for decades. Everybody believed him. He'd won a Nobel Prize after all. Who's going to argue with a Nobel Prize winner? Well, Hayflick argued. He noticed that his own cultures of normal human cells always stopped dividing after a certain number of passages. Always, no matter how carefully he prepared them, no matter how perfect the conditions. And he was brave enough to say, maybe the cells aren't failing because of something I'm doing wrong. Maybe they're failing because that's what they're supposed to do. He published his findings. People were skeptical. It took what he later described as 10 or 15 painful years for the scientific community to accept what he'd discovered. Torpedoing a half-century of belief is not easy, even in science. But he was right, and that maximum number of divisions, about 50, is now called the Hayflick limit, in his honor. In 1971, a Russian theorist named Alexei Olovnikov proposed that the mechanism behind the Hayflick limit might have something to do with the ends of the chromosomes. He couldn't copy the very tips, he argued, and so information was being lost with each division. And then in 1984, Elizabeth Blackburn and Carol Greider discovered telomerase, the enzyme that rebuilds telomerase, and the whole picture snapped into focus. They eventually won the Nobel Prize for it in 2009. The Hayflick limit, telomere shortening, telomerase, it was all connected. It was a molecular clock counting down the divisions until your cells give up and retire. So, here's the picture. Your cells have a built-in countdown, a fuse, if you like. And every time they divide, the fuse gets a little shorter. Eventually it runs out. The cells stop renewing themselves. Tissues degrade, organs falter, you age, you die. And this isn't just human cells. The Galapagos tortoise, which can live 120 years, has cells that can divide many more times than ours before hitting the limit. A mouse, which lives about 3 years, hits the limit much sooner. The number of divisions correlates with the lifespan of the species. Evolution has tuned the clock differently for different animals. But every animal has a clock. Now you might say, wait a minute, can't the cell just rebuild the telomeres? Wait, add back what was lost. And the answer is yes, actually. There is an enzyme called telomerase that can extend telomerase, rebuild the protective caps. And some of your cells do have it. Stem cells have some telomerase activity. Immune cells can activate it. But here's the sinister part. Do you know what else is very active telomerase? Cancer cells. Cancer cells have figured out how to keep their telomerase long, which is exactly how they achieve their terrible gift of dividing without limit. They're immortal in the worst possible way. So evolution seems to have struck a bargain. You can either have cells that eventually stop dividing, which protects you from cancer but means you'll age and die, or you can have cells that never stop dividing, which means cancer will eat you alive. Pick your poison, death by aging or death by cancer. The telomere system is essentially an anti-cancer mechanism that has the unfortunate side effect of making you mortal. And on top of all that, there's oxidative damage. Every time your mitochondria, those little power plants inside each cell, burn fuel to make energy, they produce reactive oxygen species, what people sometimes call free radicals. Tiny molecular shrapnel that bounces around inside your cells, nicking your DNA, damaging your proteins, oxidizing your membranes. It's like running a factory that produces excellent products but also sprays sparks everywhere. Some of those sparks hit things you'd rather they didn't. Your body has repair systems, antioxidant defenses, proofreading enzymes. Teams of molecular mechanics constantly patrolling, fixing the damage, clearing out the debris. And they're very good, astonishingly good actually. But they're not perfect. Over time, mistakes accumulate. Damage that doesn't get fixed piles up, proteins get cross-linked and stiff, DNA mutations build up in cell after cell. The pattern, that is, you gets noisier and noisier, like a photocopy of a photocopy of a photocopy. Each generation of copies a little less sharp than the last, and eventually the noise overwhelms the signal. Denham Harman proposed this idea back in 1956, what became known as the free radical theory of aging. It's been refined and complicated since then, and it's not the whole story. But the basic insight is sound. You are being damaged from the inside by the very machinery that keeps you alive. The engine that runs you is also slowly destroying you. There's a kind of tragic poetry in that if you're inclined to see it. So there it is. The physics says disorder increases. The biology says your cells have a timer. The chemistry says damage accumulates. Death is not just likely, it's inevitable. It's built into the fundamental architecture of what you are. You can eat well, exercise, take your vitamins, avoid smoking, get enough sleep. But in the end, the second law seems undefeated. It always wins. Except there's this jellyfish. In the early 1980s, two young scientists, a German named Christian Sommer and an Italian named Giorgio Bavistrello, were doing field work in the Mediterranean. They were collecting hydrozoans, which are tiny, delicate members of the jellyfish family. Small stuff. The kind of work where you wait around with glass jars and a pair of very patient eyes. They collected some specimens of a species called Turritopsis, brought them back to the lab, put them in a jar, and waited for them to grow. Now, the normal jellyfish life cycle goes like this. You start as a larva, a tiny swimming thing called a planula. The planula settles on the ocean floor and grows into a polyp, which is a little stalk-like creature attached to a rock or a shell. The polyp then buds off free-swimming jellyfish called Medusae, which is the bell-shaped form you'd recognize. The medusa grows up, reproduces, and dies. Larva, polyp, medusa, death. One way, one direction, just like everything else in the universe. But when Sommer and Bavistrello checked their jar, they found something that shouldn't have happened. Instead of maturing and dying like sensible jellyfish, some of the medusae had fallen to the bottom of the jar and turned back into polyps. Adult jellyfish had reverted to their juvenile form. That's like a butterfly turning back into a caterpillar. That's like a frog ungrowing into a tadpole. That's like me turning back into a baby. That is not supposed to happen. The arrow of development, like the arrow of time, like the arrow of entropy, is supposed to go in one direction. They stared at those polyps, they checked and rechecked. They looked for contamination, they wondered if somehow new larvae had gotten into the jar. But no, the animals were the same, the DNA was the same. The adult had become a juvenile. Development had gone backward. It took a while for the scientific community to pay attention. The discovery was presented at a workshop on hydrozoans in Italy. A marine zoologist named Stefano Pirino was there, and he later recalled the astonishment in the room. He called it counter-evidence against the fundamental dogma of biology that lives go in one direction, from fertilization of the eggs to death. Counter-evidence is a polite word. In plainer language, what they were looking at should have been as impossible as a scrambled egg unscrambling itself. And for a long time, not many people noticed it was a niche finding in a niche field. Hydrozoan biology isn't exactly front-page news. But slowly, steadily, the implications began to sink in. The species was eventually classified as Turritopsis dohrnii, and the press gave it an irresistible nickname, the immortal jellyfish. Let me describe this animal to you, because it matters that you understand how small and how modest it is. Fully grown, Turritopsis dohrnii is about 4.5 millimeters across. Oh, smaller than a pencil eraser. Smaller than the nail on your little finger. It's translucent, almost invisible in the water, with a bright red stomach showing through its bell like a tiny glowing heart. It has maybe 8 to 90 tentacles, depending on where in the world it lives. Tropical ones have fewer. Temperate ones have more. No brain, no lungs, no dedicated organs for what it does. It is by any reasonable measure among the simplest animals on Earth, and it does something that nothing else in the animal kingdom can do. When this jellyfish encounters stress, injury, starvation, disease, or even just old age, it can reverse its entire developmental program. The adult medusa sinks to the bottom and begins to transform. Its cells undergo a process called transdifferentiation. This is the key word, so let me make sure you understand what it means. In a normal organism, cells are specialists. You have muscle cells, and nerve cells, and skin cells, and liver cells. Once a cell has been assigned its job, it's committed. A muscle cell doesn't become a nerve cell. A skin cell doesn't become a blood cell. That's the basic rule of development. Cells differentiate, they specialize, and they stay specialized. That's the deal. Transdifferentiation breaks the deal. In Turritopsis dohrnii, adult specialized cells can transform directly into completely different types of specialized cells. A muscle cell can become a nerve cell. A nerve cell can become a skin cell. It's not that the cells go back to some undifferentiated stem cell state and then respecialize. No, they jump directly from one identity to another. It's as if a plumber could on a moment's notice become an electrician, not by going back to trade school, but by simply deciding to be one and having all the knowledge instantly. And through this process, the adult jellyfish, with all its mature specialized tissues, reorganizes itself back into a polyp, a juvenile. It has effectively rewound its own biological clock, and from that polyp, new jellyfish will eventually bud and grow, genetically identical to the original, the same animal, reborn, not a copy, not offspring, the same individual, starting over. A Japanese scientist named Shun Kubota at Kyoto University managed to keep a colony of these animals alive in his laboratory for an extended period, something extremely difficult to do. These are finicky creatures. You have to inspect them every single day. You have to make sure they've properly digested their food, tiny brine shrimp cysts. The water conditions have to be just right. Almost nobody else has managed it. But Kubota did. And he reported that during a 2-year span, the colony reversed itself 11 times. 11 times, falling back, growing up again, falling back, growing up again, over and over. Theoretically, there is no limit to how many times this can happen. And the resilience is extraordinary. Researchers have tried starving these animals. They've heated them, they've exposed them to ultraviolet light. They've cut them with instruments, insults that would kill virtually any other animal, and the jellyfish simply shrugs, sinks to the bottom, and starts the reversal process.